Hello everyone, I'm Dr. Tushar Pandey and I'll be discussing today the recommendations that have come in the EST 2022 book for the management of resistant as well as intolerant chronic myeloid leukemia cases. So, I've divided the presentation into four parts. Number one, the basic history, followed by molecular basis, third being the highlights from the update, and fourth being case scenarios. So, the references of various slides are mentioned in each slide, as well as in the basic history. I would like to share that I have taken it from Pathology Doodles, and while the highlights and the case scenarios are from the EST 2022 book. So we'll start. So in 1959, Dr. Novell and Dr. Hungertard in Philadelphia found that it is, in cases of CML, there is an abnormality present in the chromosome 22, and later it was named as the Philadelphia chromosome. Then, can anyone identify who this lady is? She is a very important figure when it comes to leukemias. She is Dr. Janet Rowley, and she discovered in the 1970s that it's a translocation 821 that causes the RUNX1T1, as well as the translocation 1517 that causes APML. And in addition, and she had also found that translocation 922 is the root cause of the Philadelphia chromosome, the derivative chromosome 22. In the 1980s, Dr. Nora Hesterkamp cloned the name and named the BCR, and she showed that due to translocation 922, the genes BCR and ABL1 are rearranged and they cause CML. Dr. Owen White then discovered the role of ABL1 as a tyrosine kinase, and this helped to understand how BCR-ABL1 derives its pathogenesis. In the 1990s, then imatinib was discovered by these two doctors, Dr. Brian Drucker, an oncologist, and Dr. Nicholas Lydon. So to enhance its functionality, they modified it by adding methyl groups as well as benzamide groups. They had begun their phase 1 trials in 1998 and achieved FDA approval in 2001 for the use in CML. Starting with the epidemiology. So when it comes to CMLs, the worldwide annual incidence is about 1 to 2 cases per 1 lakh population. It can occur at any age, with the incidence increasing with the age. It comprises about 15 to 20% of all leukemias. The median age at diagnosis is 5th to 6th decades, and there is a mild male preponderance. This data has been taken from the WHO 2016 Blue Book. Now, the Indian data taken from this article mentioned here in the Journal of Pediatric Oncology in 2013. CML was found to be the commonest adult leukemia in the Indian population, accounting about 30 to 60% of all the adult leukemias, with a male preponderance and a median age of about 32 to 42 years. And it is not only for CMLs, for other conditions also like MDS and various other malignancies. We are a decade ahead of our European and American counterparts. So while the median age is about 55 years in Europe and 66 years in America, the median age of presentation for CML is about 32 to 42 years. Now moving to molecular pathology. So using the quinacrine and GIMSA bending, Dr. Rowley reported in 1973 that material missing from chromosome 22 was not actually lost or deleted from the cell, but was actually translocated to the distal portion of long arm of chromosome 9. The amount of material translocated to chromosome 9 was equivalent to that amount lost by chromosome 22, and the translocation was predicted to be a balanced translocation. Moreover, breaks were localized to band 34 on the long arm of chromosome 9 and band 11 on the long arm of chromosome 22. Hence, the classic Philadelphia chromosome is translocation 922, Q34 and Q11, abbreviated as TPH. Now, in this photograph, I am just showing you that this is the chromosome number 9 in green, and the blue one is the chromosome 22. The 9 contains ABL, and while 22 contains BCR. Now the breakage occurs in chromosome 9 as well as chromosome 22, and this ABL domain of chromosome 9 binds to the BCR domain of chromosome 22. Hence, the point that I want to stress here for our young budding doctors, physicians and hematologists and hematopathologists that it is the derivative chromosome 22, means chromosome 22, that is the BCR present, BBL is getting bound to dead BCR in chromosome 22, and it is the abnormality is present in the chromosome 22. Hence, it is a derivative chromosome 22, which is known as Philadelphia chromosome. So, why native ABL1 has a tightly regulated kinase activity, whereas the BCR ABL1 shows a constitutive activation? Well, the reason lies in that BCR ABL1 loses the N terminal or the N cap, which contains sequence for merostylization or merostylation. So the N-terminal meristic acid group binds to a deep hydrophobic pocket which is present in the C-terminal of the kinase domain. Interaction of this merostylated N-cap with the C-terminal lobe is critical to maintain the auto-inhibited state, that is to restrict its activity. So loss of this region and fusion with the BCR sequences encompassing the oligomerization domain and Y177, they evade the physiological control of the kinase. So in this figure, the cap actually domain, this is in the native ABL1 kinase enzyme, and this cap contains various sequences for merostylation. So what it does, it keeps the configuration in a closed loop or a closed configuration, and the enzyme is rendered inactive. So when the translocation 922 occurs, so this cap with sequences for merostylation is replaced by BCR ABL1, which contains sequences for oligomerization, and an open conformation is formed, which makes the enzyme very active, and ATP binds to the catalytic site, and phosphorylation of various substrates like JAK2, STAT5 occurs, which promotes the cascade of events which ultimately leads to leukemogenesis. So basically there are three steps in the mechanism of ABL tyrosine kinase activation. ABL is maintained as we have told, maintained in a close inactive conformation by a mechanism that is known as latching of the N-terminal merostyl group. So this latching of this N-terminus of ABL to its kinase domain places this SH2 and SH3 domains of the ABL on the back side of the large and small lobes of the kinase, respectively clamping the structure together, as we can see here. Right in the leftmost figure, this merostyl pocket is active and it is making clamping. So to render the enzyme active, an unlatching phenomenon and unclamping phenomenon has to occur, and this unclamping is done with the help of certain phosphotyrosine and as well as proline rich proteins which bind to the SH2 and SH3 domains respectively and open up the kinase and expose the activation loop outwards, leading to tyrosine 412 phosphorylation and full activation of the kinase, as we can see here. So ultimately tyrosine 412 phosphorylation is occurring and this cascade of events is propagated. Here again, this is taken from a video by Gleevec, where the company they were showing how the drug acts. So I've taken a glimpse of it. So basically ATP binding occurs in the BCR-ABL1 fusion protein, and it causes phosphorylation of the substrate, which could be JAK2, STAT5 and other, and leukemogenesis is initiated. So summarizing the events, BCR-ABL1 enhanced tyrosine kinase activity is present because of it. Constitutive activation of several signal transduction pathways occurs in leukemic phenotype, which ultimately leads to deregulated proliferation, defective apoptosis in response to the mutagenic stimuli, and reduced adherence of the cells to the bone marrow stroma. Okay. Now moving to mechanism of action of drugs.